Hello everyone. Today we'll be talking about the neuroembryology of the spinal cord and its clinical anomalies. I am Dr. Sonyaolu. As a way of introduction, the spinal cord actually develops from the caudal part of the neural tube. If you remember, during the embryology of the brain, I'm sure by now you've been taught the embryology of the brain. During the embryology of the brain, we spoke about the formation of the neural cord, the neuropod, which closes cranially and caudally. And the closure actually begins from the cervical region. And so we are looking at how the spinal cord develops from the neural tube. So the caudal part is actually what forms the spinal cord. Because of the relationship between the spinal cord and the vertebral column, their development is also linked. And once there is a problem, or if there is any problem with the differentiation process, which we'll be discussing later on, or the closure of the neural tube itself, it can result into a congenital anomalies. Before the end of this lecture, we'll be discussing some of the congenital anomalies that are associated with any of these defects. Let's talk briefly about the formation of the neural tube. And this is actually supposed to have been taken in the previous lecture, but we'll do a little bit of recap. We have the epithelial layer, the epidermal layer being induced by the somite, and it results into the formation of the neural plate, which later folds to become the neural fold. Then it closes up to become the neural tube. This neural tube, which becomes the neuropod, closes at the cranial region and caudally. The cranial part closes at about 25 days embryonic life, while at day 27 we have the entire closure, the complete closure of the caudal neuropod. It is this caudal neural tube that becomes the spinal cord. So this is just like a recap, just as I've said earlier. Now let's move on to the formation of the spinal cord proper. Once the neural tube closes, or the caudal part of the neural tube closes up, the neuroepithelium that lines this neural tube begins to proliferate, meaning that they increase in number as they divide. So this proliferation results into the formation of a three-layer. We have the ventral zone, we have the mantle zone, and we have the marginal zone. The marginal layer is that part that is found at the outer region, while the mantle, it's the inner part, which you can see from here. Just a minute. Okay. So you have the mantle, which you can see here. You have the marginal. So the ventral zone will eventually have some populations of cells, which are the neuroblasts, which will eventually become neurons, and the glioblast. You have another population of cells, which are referred to as the glioblast. All right, so the mantle zone will become the gray matter, and we also have them as the neuronal cell body. The marginal zone is the white matter, which consists of the nerve fibers and the nerve fibers. All right, so let me just use this diagram to explain this better. So you have the neural tube form, you have the neuroepithelium proliferating, dividing to divide these into different layers. These layers will eventually be grouped. Some will be at the roof area, and some will also find themselves at the floor region, while some will be at the middle. Those present at the floor region will form the halo plate, and those at the base, at the floor, at the roof will form the hala plate, and those at the floor will form the basal plate. These two plates are separated by a groove in the middle, which I will show you. Let me just show you that briefly. By this groove, by the groove in the middle, and this is called the sulcus limitan. These divide the plate into hala plate and the basal plate, and in the middle you have the central canal. Okay, so that is just about the differentiation of the spinal cord. However, this differentiation which forms the hala plate and the basal plate does not just occur. They are controlled by another tightly regulated system, which is more molecular. It has to do with the expression of genes. So there are certain genes that will be expressed that will determine the movement of the cells. So based on the type of gene that is being expressed at that particular moment or in that particular region, that might determine where the cell moves to, either to the hala, to the middle, forming the gray matter or forming the white matter, depending on what particular switch is being turned on. I won't be discussing the details about this gene, but it is just for you to understand that this movement that we talk about did not just occur. There is another system that controls this movement. So the hala plate, which is found at the dorsal region, will eventually form the sensory, make up the sensory neuron, while the basal plate, which is at the ventral part, will become or form the motor neuron. And in the middle, you have some group of cells which are called the intermediate zone, and these will become the autonomic neurons. Okay, so let's discuss the spinal nerves formation. If you remember, we've discussed how the sensory neurons and the motor neuron is formed. So the axons of the motor neurons will exit from the basal plate, and this becomes the ventral root, just as you've seen here. So this is the ventral root. The motor axons from the basal plate will exit through this ventral root, while the sensory neurons, which arise from the neural crest cell, if you remember when we're discussing the formation of the neural tube, we spoke about the neural crest cell. So this neural crest cell forms the dorsal root ganglia, and the axons of this ganglia will enter into the hala plate, just as you can see here. It will enter into the hala plate, and this becomes the dorsal root. When the dorsal root and the ventral root meet, they form the spinal nerve. Okay, and you can see that also from the diagram below. You can see how the nerves either exit or enters into the basal, leaves the basal plate or enters through the hala plate. All right, so let's discuss the growth changes. During early embryo, the spinal cord seems to be at the same length as the vertebral canal, meaning that it extends the entire length of the vertebral canal during early embryonic development. But at birth, these changes simply because at this particular point in time, the vertebral column tends to grow faster than the spinal cord. So the spinal cord will end at L3, while the vertebral column continues. While in male, as the vertebral column seems to grow, the faster it becomes, the vertebral column grows much more better in adult, has grown much better in the adulthood, and so you have the spinal cord ending at L1, L2. This is what actually brings the disparity between the spinal cord during early embryo birth and during adults. The disparity due to this fast growth of vertebral column will result into the caudal equinus, the caudal equina. So these are the groups of nerve fibers that is located at the base of the spinal cord. Let's talk about the myelination. The myelination does not begin until late fetal period, which eventually continues even after birth. So the ascending tract neurons tend to be myelinated earlier compared to the descending tract, and because of this, for babies, they tend to have more reflex. But as they grow after birth, the motor coordination will gradually be developed. So the motor coordination will improve gradually after birth as the myelination process occurs. Just because we said that the myelination process does not really begin until late fetal, which then continues after birth. So let's talk about the clinical anomalies that can arise as a result of development of the spinal cord. We begin the development of the spinal cord by talking about the neural tube. So there are some times where there are defects in the closure of the neural tube, leaving a gap in the vertebral column. Don't forget I said that the vertebral column development is closely linked to that of the spinal cord. So failure, or if there due to an opening in some area of the vertebral column, it could result into spinal bifida occupta. This actually is a very common anomaly, but people don't even know they have it, simply because it is asymptomatic. But they can get to know by sometimes through dimple on the back, seeing dimple at the back showing a kind of depression, or at times stuff of air there could be an indication that there is a spinal bifida occupta that has occurred. All right, so the next one is the spinal bifida cystica. The difference between the occulta and the cystica is, for cystica, there is actually protrusion of the meninges out of that region that was not completely fused, so thereby causing the meninges hernia. The third type is the myelomeningocele, which is a more severe type, which is a more severe one because it causes severe neurological deficit. And this involve the protrusion of the spinal cord along with the meninges in that region that was not fused. And you can see that from the diagram that is shown. Other congenital anomalies that we'll discuss is the syringomyelia, which is as a result of defect in the cavitation in the spinal cord. This usually occurs around the neck region, the cervical region, and then there will be like a loss of sensation around the shoulder to the hand, forming like a cape pattern. So there will be loss of sensation of pain and temperature at that particular region, and it is as a result of abnormal canalization of the spinal cord at this region. We have the tetheral cord syndrome, which arise as a result of a shorter or a low conus medullaris. Conus medullaris is that cone-shaped region that forms at the inferior part of the spinal cord. So when it is shorter than when it is abnormally low conus medullaris, when it is abnormally low. So what happened is that it results into a situation whereby one is not able to control their bladder, and therefore it results into incontinence, and also there is leg weakness. Moving away from that, we'll go to dystomylia, which has to do with the splitting of the spinal cord, and this splitting can be by separation through a bony structure or through a cartilaginous septum, as you can see in the illustration that is on this slide. Okay, last that we'll be discussing regarding the congenital anomalies is the caudal regression syndrome, which occurs usually, is said to be associated with maternal diabetes, and because of that there is incomplete development of the lower spine and the spinal cord, resulting into a situation whereby the lower region is not well developed, and therefore it causes weakness in the leg, and also some kind of other abnormalities can also occur as a result of this. So in summary, we've discussed the spinal cord being formed from the caudal neural tube, and also we spoke about the differentiation as a result of the proliferation that occurs, proliferation of the neural epithelium that occurs, thereby organizing this proliferated cell. This proliferated cell now become organized into layers of cell, into layers. We have some at the marginal region, then we have some which are the zone, the mantle zone, and also the ventricular zone. These cells are now split into two plates, which forms the roof and the floor, and is separated by a sulcus limitans. The floor, which is called the basal plate, and the roof, which is called the alar plate. And we also spoke about the disproportionate growth of the vertebral column and the spinal cord, and how it's also explained. It is also explained by the conus. It is also explained by the conus medullaris and the caudal equina. All right. So lastly, we spoke about the anomalies that occurs as a result of defect in this region. Thank you.